Oh, okay, good. You have the live. Let's see if the live stream is working before we proceed. Sure. Excellent connection. Okay, I see it.
It's working.
Did you open it up on YouTube?
Yeah. I just had it up and it was working.
Okay, awesome. All right. Be like 30 more seconds.
Yeah.
People are still coming in.
Yeah.
P is lower compared to Uber.
Okay, so thank you everybody who's able to join us today. As usual, the procedure will be fairly straightforward. In that, like, if you want to interrupt and ask questions, you are definitely welcome to, or you can kind of raise your hands with the hand emoji and we'll try to stop Sheila and ask, so you can ask. Yeah, so we're honored to have Shiladitya Banerjee present today. Sheila did his bachelor's in physics at Chennai Institute in India and moved to the US for a physics PhD at Syracuse, which he did. He was advised by Christina Marchetti. He worked on a number of problems at the interface of soft matter physics and fluid dynamics, working at the intracell scale, determining how like active gels like the actin cytoskeleton could make contractile forces, how at the intercellular scale and the how cells interacted with each other and the extracellular matrix. And he was very prolific as a back then and also now. He came to Chicago in 2013 as a Kadanoff-Rice postdoctoral fellow. That's where I was fortunate to meet him and work together on a lot of agent-based models and stimulating the actin cytoskeleton. And he was an amazing mentor who I could always turn to for math questions, physics questions, computing questions. He was extremely instrumental in my own career. In 2016, he moved to University College London as a junior lecturer. He was very quickly kind of promoted through their ranks, but he actually moved back to the US to come to Carnegie Mellon, Carnegie Mellon's physics department, where he currently is, and in 2020 got there. And he's been a lot of fun to work with personally, and I'm very excited to hear him talk about bacteria and their how they work in different environments. So yeah, I think you can take it away, Sheila.
Thanks a lot, Simon, for the kind introduction. So I'm happy to be here talking to theorists, which these days I don't get enough opportunities to. So my talk today will focus on understanding the interplay between physiology and morphology, okay? And how do we connect physiological properties of a cell, such as a microorganism, bacteria, to its shape, and how do we understand it in terms of optimizing resources that an organism has, right? I should first acknowledge that most of the work I'll talk about is done by my former postdoc, Nicola Ojkic, who was with me at UCL. He was in fact a postdoc with me till early this year, and Diana Sarbanescu, who is also who is my current PhD student, working at UCL. So first of all, please feel free to interrupt me whenever you want. So what do I mean by interplay between morphology and physiology? So let's give you a very simple example. So you can watch a bacteria grow and divide and replicate, okay, and watch them replicate exponentially, right? And using that, you can compute the rate of growth of that population of cells, right? And if you measure the size of individual bacterium, what you find is a very striking positive correlation between cell size and growth rate, right? So the morphology of cells is intimately connected to the rate of growth, okay? And this is an instance of a law, right? A phenomenological law that we would like to understand. It's one of the things we'd like to understand, but I wanted to start with it because it's a very simple law, but the underlying mechanism is not so simple as we'll see, okay? And this is inherently a multi-scale problem because, well, what we're looking at, so the population scale growth rate of a population of bacterium depend on how fast do individual cells elongate, right? And that ultimately depends on how fast cells synthesize proteins, right? So the challenge, I guess, in physics or systems biology is to connect the scales, right? And we would try to do that using a top-down approach that, okay. So the type of questions we will address in this talk, I'll try to address 4 questions. I'm not sure if I'll be able to answer all of them, but the first one is how do cells couple growth and division to regulate their size, right? So size is one of the most important physiological properties of cell, aside from growth rate. How do cells allocate resources to regulate size and growth, right? Then cells need to consume food and transform them into amino acids, which needs to make proteins and ribosomes, right? So they need to have some optimization strategies, which we would like to understand, right? Thirdly, what is the connection between the geometric form of a cell and the fitness, right? Fitness for growth and replication, okay? And lastly, how do cells adapt to changing environment, right? So if you have an organism whose steady state properties you understand well, how do you understand their adaptation and response to changing environments, right? And so most of the open questions, I think, lie in that, in the last part, sort of adaptation to changing and fluctuating environments. So, okay. All right, so, let me start with the nutrient growth law, which I already alluded to. So the nutrient growth law basically states that the cell size of the average size of a cell increases exponentially with the growth rate. So this is a log linear plot of cell size, and this was actually discovered about 6 decades ago now, right? By Schechter et al. And this was done by experimenting cells in different nutrient media, right? So as you increase the nutrient concentration of media, cells grow faster. And what they saw is that the cell size also gets larger. It's important to note that this is average cell size because cell size is always dynamic, right? So cells elongate exponentially during the cell cycle, okay? So this is an observation that was made during balanced growth conditions, okay? So what do I mean by balanced growth is that all the extensive properties of a cell, such as its mass, DNA abundance, or RNA abundance, all of them sort of grow at the same rate as a cell, right? And so the nutrient growth law was first, I would say, discovered at the population level. Like, so you're looking at a population average cell size, population average, sorry, the growth rate of that population. But recently, using Mother Machine, which you see on the top left, sort of a movie of a Mother Machine developed by Sukhjin Jones' lab at UCSD. And they studied bacterial growth in different growth media at a single cell level. And they found that the nutrient growth law still holds, right, on average, right? Although you observe significant fluctuations in growth rate and cell size at the single cell level, but on average, they kind of follow that same curve, right?
Can I ask a question?
Yes.
Does this law just mean that you're dividing after the same period, even as the growth rate change? Is that?
No, so the time period of growth depends on the growth rate. So in fast-growing medium, cells divide at a much shorter time. Okay.
Mhm.
Does that answer your question?
Yeah. I just trying to understand like geometrically what the law means.
Oh, geometrically, the law means, I mean, it's pretty simple. I mean, I haven't yet gone into the details of the cell cycle yet, but just in fast-growing medium, when cells grow faster, right, they are larger in size. That's what it means on average. And of course, the cell size is dynamic, right? So here, for example, they're plotting newborn cell size at birth, right? You can also do a similar correlation between cell size at division, right? Okay, cool. So all of this is good, but the surprise was that, sorry.
Sheila, can I just ask a question?
Yes,
So the blue, within one nutrient, so TSP or glucose or whatever, just so that I understand, the blue points are if I took Mother Machine and I increased the concentration of glucose, and then I see within, yeah, I just want to understand.
Okay. No, so so let's look at the curve for synthetic rich, for example. So in that case, they're not changing any nutrient concentration or anything.
Okay.
This is the intrinsic fluctuation of a cell.
But this is the intrinsic fluctuation.
Okay.
Thank you.
So
Thank you.
okay, cool. So the nutrient growth law holds under nutrient perturbation, but the surprise was that this breaks down when you inhibit translation. Okay. So that's quite important. So how fast cells grow depends on translation, right? So how fast translation occurs, proteins are synthesized, right? And you can use antibiotics to bacteria to sort of inhibit translation. And what you see is a mess, right? So the nutrient growth law breaks down immediately. And there is no general trend here, okay? And if you read this paper by Fang Wei Si et al., you'll see that they find a way to rescale this curve. I'm not going to get there, but the question I want to address is what is the molecular origin of the cell size control, okay? And what is the connection between cell size, protein synthesis, and growth rate, okay? So it's not too simple because obviously there's a lot here, okay? To sort of address that, let me dive into what's happening at the single cell level, right? And sort of go back to the mother machine data. So here you see a movie of the mother machine where cells grow in dead and micro channels. The width of the channels is slightly larger than the width of the cell, right? And cells grow in a line, and as they reach the top, they face the flow and they're sort of washed away, right? So you can image the growth of E. coli, and you obviously find that cells, so if you follow the trajectory of a mother cell through birth and division, you find that cells grow exponentially, right? What was discovered by, I would say, by Sakjun's group and others as well around the same time in different organisms, we in fact worked on a different organism, arrived at a slightly different conclusion. But mostly, I would say that, so these cells control their size by an adder mechanism. Okay, so what that means is cell divides after adding a fixed amount of size, okay? Statistically speaking, what this means is that the added size shows no correlation with the birth size of that cell, right? So, so this is called the adder model, right? This is quite different from other types of models that are quite popular, such as Sizer, where cells divide at a fixed size, or cells divide, or a timer model where cells divide after a constant time period. But here, the adder model ensures convergence to homeostasis, okay? Which some of the other models don't. Sizer model does, but one of the great features of the adder model is that it ensures robust convergence to the mean size. So if we have fluctuations away from the mean size, the cell size converges rather quickly within a few generations to its mean size, right? So at a single cell level, cell size is controlled by this adder mechanism, right? which means that cells grow by a fixed amount, then divides, okay? And in the next cycle, they also grow by a fixed amount. And that fixed amount does not depend on the cell size at birth, right? Cells could be big or small, but they add the same amount, which means that bigger cells have a much shorter cell cycle than a smaller cell, okay? There was a chat. Okay. Okay.
To be clear, the reason that they converge is because they always divide in half, not divide at that extra amount added, right?
So even for asymmetric division, it ensures convergence. So they don't need to divide in half. They can divide in 60-40.
Okay, no, all right. But that ratio doesn't really depend on the delta that's being added.
No, so the delta is independent of the ratio.
Okay.
And what I want to say is that the final size will depend on whether you're asymmetric or not, but this convergence doesn't depend on asymmetry. So in fact, we studied.
Can I ask dumb questions?
Yeah.
So the adder model is telling me something about cell division, right? And then why? But ultimately, you're interested in the size of the cell at right, just before it's divided, or statistically maybe the size of all the cells. Why is it clear why those two things are connected?
So what the adder model tells you is that the cell size at birth of a population will converge to the added size delta, okay? And the average size of a cell in a population, so all the cell cycles may not be synchronized, but the average, so if you compute the average size of a cell, and you will see that it is proportional to the birth size of a cell, right? With some factor of log 2, etc. So so the average size of a population is proportional to the added size, okay? And you can do a simple calculation to sort of see that, but okay. So
okay.
Not all organisms are adders. I just wanted to make that point. I don't want to get into the details here. For example, many of the organisms, at least E. coli, bacillus, budding yeast, they are sort of adders. I mean, we study something called Caulobacter, as highlighted here when I was a postdoc, and it's sort of in between an adder and a timer. But anyway, but let's.
Sorry to interrupt. Can I ask just a clarification question on the different types of models?
Yes.
So in principle, like pragmatically speaking, what is the difference between an adder and a time model? So if I look at your plot of this exponential dependence of size, right, on the growth rate, like a simple kind of naive model would be that there's some fundamental rate limiting process a cell needs to do in order to divide. And it just takes a certain amount of time or a certain amount of, and I'm assuming that these data are all in like rich media, right? So that there's no, so there's controlled out for the medium. So if that's the case, then what is the difference between, so like both the time as well as the adder model could be just a proxy for needing to do something, right?
So the problem with the timer model is that it doesn't lead to convergence, it leads to instability. So if you have a model of exponential growth where you say that the cells divide after a fixed amount of time, okay, which essentially means that cells double in size or once they divide. So this has been sort of the canonical model that biologists have used for a long time, right? So this model is wrong. It leads to instabilities and sort of divergence of size in a population. So your question was, what is the fundamental difference between a timer and an adder? The adder ensures size convergence, timer does not. And timer means that cells grow, so it depends whether cells grow linearly or exponentially. So for exponential growth, timer means that cells grow as a fixed multiple of the initial size. And the adder means that you add a fixed amount of size, okay? There are no known organisms which are timers, okay? Most of the organisms are either adder or weakly adder, or more towards a sizer where they divide at a fixed size. Okay, so fish and yeast is a good example of a sizer.
Yeah, I guess my question is more in terms of the observer. So as an observer, right, if I see that a cell, and this is, let's say, all in rich media, a cell always divides after it grows an extra amount, like that would also be like it'll always divide after a certain fixed amount of time too, right?
No.
So like under what circumstance would those two things, because if it's just like whenever it divides, however big it is, it just needs to like grow this extra amount, then why is that growth of that extra amount taking different times? I'm just trying to understand as an observer how to distinguish.
Because they're growing exponentially. So if you're growing exponentially, if you add a larger amount, so what you're saying is only true if it's linear growth. If it's linear growth, then adding a fixed amount of size means you grow for a fixed amount of time. Okay. For exponential growth, these two are not the same. Okay.
Okay, I see. So I guess like here, because I'm thinking about one splitting, it's just like there's just a certain amount of, like in the movies that you show where a cell starts and it's divided now, and no matter how big it is, if it grows a certain amount, right, and no matter and that growth could be in whatever time dependent way, but once it grows a certain amount, it will divide, right? So if I just, even though in that time process it's not a linear in time, there's still a fixed time it takes for it to divide.
No.
Right.
So that time interval will vary depending on your initial size. Okay. It's
okay. I see.
It's actually quite simple math you can do at home, and let me just move on. So and we can come back if still there is question. So okay, so all of this was well established maybe 5 years ago or so. So we wanted to think deeply about the molecular model for it, right? So what's happening at the molecular level, like what is the size sensor, right? So we developed a model called the threshold initiation model, and I just wanted to talk briefly about it. And this sort of lies at the how to connect protein synthesis with cell size and division. So the model works like this. So imagine the cell starts to grow, right? When it's born, so it synthesizes something called division proteins X, right? And these division proteins are synthesized at the same rate as the growth of cell size. Okay, so divisions protein accumulate as the cell grows. Okay, and cell divides when a fixed threshold of this division protein is reached. Okay, so it's very simple. So at the start of the cell, let's say at the start of the cell cycle, you have 0 of this proteins. Okay, it doesn't have to be 0. There could be some initial condition, but it grows at the same rate as cell size, and then the cell divides once you hit a fixed threshold. Okay, and these proteins are, so the rate of growth of this protein, so the rate of synthesis of the protein is Kp, and the rate of growth of the cell is K, right? And these proteins are also degraded at some rate mu. Okay, which is, I mean, true for all proteins, right? So yeah, so this is the basic model. It's called threshold initiation model, right? So this model was inspired by these experiments that were done a couple of years ago by Fang Wei Si, where they find that the bacterial division protein FTSE reach a threshold at the time of cell division, right? And by threshold, I mean the amount of the proteins, not the concentration. The abundance of these proteins reach a threshold. Okay, and this could be measured by measuring sort of the integrated intensity of the rings that FTSE form at the bacterial meat cell, right? And so you can do a simple math like solving these problems, and you can show that this model leads to adder behavior. Okay, if these proteins are degraded much slowly than the rate of growth. Okay, which is true for FTSE at least, right? So what you find is that the amount of added size between cell cycle is constant for a given media. Okay, for in a given medium, given growth medium, the growth rate is constant. Cells are reaching a constant threshold, and the rate of synthesis is also constant. So given all of that, so cells reach a fixed, so cells grow by a fixed amount within cell cycle, right? And in the other limit, where the cell is growing really slowly, like not in a rich medium, cells behave more like a sizer where the division size is constant. In fact, it has been seen for E. coli that if E. coli grows in a very slow medium, they're not adder-like, okay? So this model kind of behaves both like an adder and a sizer, but let's focus on this regime where cell grows at a much faster rate than the rate of degradation, which is true for most cells, and then you can sort of compute a simple expression for the average cell size, which is basically the ratio of the rate of growth and the rate of synthesis of this division proteins, okay? So this is a renormalized constant Kp, where we have absorbed most of the constants, but the basic idea is that rate of growth of a cell depends positively on growth rate divided by the division protein synthesis rate, okay? So what we have done is we have related size to two rate constants, okay? Growth rate and the rate of protein synthesis, right? So now it becomes a dynamic quantity, right? So with this, we can try to understand what happens. So how is Kp regulated, right? How is rate of protein synthesis controlled in different environments?
Shila,
So,
can I ask a question, please?
Yes.
It's actually very beautiful. I like how it interpolates, the asymptotics interpolate very smoothly. So it's very elegant. The term Kp, kappa PV, the hypothesis that the production rate is proportional to volume.
Yeah.
This is of course a hypothesis that the production rate. So I get it, but I'm just saying, if one did look at some proteomics or some sort of protein level data, are there some classes of protein that fall into this bracket?
Yeah, I mean, we believe that FTSC does.
I see.
And if you measure in the single cell experiments, if you measure the intensity of the proteins, and you see that it linearly correlates with the cell size. Okay, so the Kp is a volume specific rate, so it means that the cells are synthesized within the cytoplasmic. Sorry, these proteins are synthesized within the cytoplasmic volume, and that's how it comes actually. Yeah.
Beautiful.
Okay. So we wanted to understand how Kp is regulated, because that lies under the control of volume, right? So K is controlled by, this kappa is controlled by nutrient growth media. So what we found was very interesting. And so we estimated Kp from experimental data. And we found that there is a negative correlation between Kp and K, right? Which underlies some sort of trade-off between the rates of growth and rates of division protein synthesis, okay? So as cells are growing faster, it is synthesizing protein slower, okay? So there is some sort of inherent trade-off. And what this means, so, let me get into what it means, but if you use this relationship, so this linear relationship, and plug it into this simple expression, you can capture the nutrient growth law very well, okay? In fact, it is not exponential. It looks like an exponential, but yeah. And it fits very well with lots of different data collected by lots of different groups. Okay. So so what is the mechanistic origin of this trade-off? So you can think about it simply as some sort of a resource allocation strategy, right? As more nutrients become available, right? Cells. So as cells grow faster, it's devoting more resources to growth and less to division, right? So cells as a, which makes the cells larger, right? If you don't have much nutrients available, right, cells are devoting more resources to division. So cells are dividing more than they grow, so they become smaller on average. Okay.
Sorry, Sheila, do you mean this as a proportion or in absolute terms? So if you're growing faster, as a fraction, less is being put into division machinery, or in some
in terms of mass fractions, let's say, So let me get into what I mean exactly. So what you can think about a simple model. I unfortunately will not get into a lot of details on this, but happy to answer. Sorry, let me just get rid of this annotation first. Okay, so how do we understand this? So this can be understood from an allocation strategy of different proteins, right? So let's think about a very simple minimal model of a cell, okay? This is a physicist model. Of course, the cell is very complex, right? So cell takes in some external nutrients, which are these food molecules represented by F, okay? And the food molecules are brought inside the cell by transporters, okay? Which are labeled by P. So P converts the external nutrients into amino acids, and amino acids then use ribosome to convert this nutrients into all different types of proteins inside the cell, including ribosomes themselves, right? Right. So so we sort of developed a sort of minimal coarse-grained model of the cellular proteome, which is made up of 4 different types of protein sectors, okay? And this is sort of an extension of earlier work done by Terry Waugh, where they were looking at a 3-component proteome model. So here we consider a 4-component model, where one of the component is a housekeeping sector, which does not change with growth medium. So this part is sort of, the sector Q is invariant under changes to translation or growth rate, okay? There's a ribosomal sector, which controls all the ribosomal proteins. There's a structural sector, which is X, which contains proteins like division proteins. And then there's a sector, which I could P, that controls metabolism, transport, etc. Right? So we can assign mass fraction to each of these sectors. So they have to add up to 1, right? And at steady state, you expect some sort of flux balance, right? So the rate of import of nutrients, right, Kn times P, phi P, must be balanced by rate of protein synthesis by ribosomes, okay? And if you impose this flux balance, what you find is that there is a negative correlation between the mass fraction of division proteins and the mass fraction of ribosomes, okay? And this underlies sort of this trade-off between growth and division, because phi X is controlling the division sector, and phi R is controlling the growth sector, because the rate of growth of a cell is positively correlated with the amount of, the mass fraction of ribosomes, okay? Okay, so using flux balance at steady state, and you can, and this minimal sort of growth model, so you can sort of mechanistically understand what's happening, right? As more resources, more nutrients become available, cells are spending those resources to growth, right? And not division, so cells are getting larger, right? So this is summarized using this plot here. So on the y-axis, we have division protein synthesis rate. On the x-axis, we have growth rate, right? And as you change the nutrient quality, what happens is that you synthesize division protein slower than growth rate, okay? So cells become larger on average. And as the new nutrient is depleted, the reverse happens, okay? So this sort of framework of resource allocation strategy was very useful, not only to understand cellular response to nutrient perturbations, but also we could understand how cells adapt to translation inhibition, right? And if you remember a few slides ago, I showed that under translation inhibition, the sort of this whole story breaks down, right? And using the model, we can actually predict what happens to cell size if you inhibit translation, which means that you block ribosomes, right? And we see that you'd see different types of trends. In nutrient-rich medium, cells reduce their size. In nutrient, if new nutrients are not available much, then cells tend to increase their size. And this has to do with actually control of surface to volume ratio that I want to get into next. What cells do is that if you're in a if not a lot of nutrients are available, right? Cells would like to increase their volume, right? So increase their size, and so we wanted to understand so what this means for the fitness of a cell, okay? And so that's one of the things we want to get into in a bit. So okay, so so this proteome allocation strategy was very useful to understand cell size regulation, right? And we wanted to also think about it from a geometric perspective, what it means, right? And if you sort of think about the growth of a rod-shaped cell, okay, and in particular for E. coli, what happens is that the length grows exponentially, but the width of a cell stays constant. So you can write the growth law in terms of the length, like the rate of change of length is proportional to the length. And then there's a division protein, Kp, which is synthesized in proportion to the length, okay? Because the width of a cell is staying constant in a particular growth media, right? Cells only grow horizontally. Okay, so the interesting thing is that, so you using this model, as I showed before, that the amount of length that you add is proportional to x naught, which is the threshold that you reach, and then the ratio of the two rates, right? Kappa over K, kappa P. So this threshold amount of division protein, so if we specifically think about FTSZ that forms a ring at the mid cell, it scales with the width of a cell, okay? Because it scales with the circumference of a cell. So this threshold amount that is reached has to scale with the width of the cell, which means that the amount of size that you add must scale with the width of a cell, okay? Which means that, you know, in different growth media, cells have different different width. If cells are growing faster, it has a higher width. If cells are growing slower, it has a much smaller width, right? So what this means is the amount of length that you add scales with the width, which means that there's a conservation of aspect ratio of the cell, okay? And there's tight control of aspect ratio across growth media. And this is sort of very well illustrated with these plots at the single cell level, where we see that the aspect ratio distribution is invariant, okay, irrespective of the growth media. So you can change the growth rate of the cell. Cells can be longer and wider, but the aspect ratio remains conserved, okay? You see a lot of fluctuations at the single cell level, but those fluctuations are uncorrelated, okay, to growth rate. So there is almost a fixed aspect ratio at 4 for E. coli, okay? So we wanted to understand what are the implications of this relationship. So it seems like longitudinal growth is coupled to the other dimension of the cell, right, the weight. And so my postdoc then wanted to look at cell shape data. So he was very fascinated with this, and he wanted to look at cell shape data. So we started collecting a lot of morphological data of bacteria, okay? And we found this a very beautiful geometric law that bacterial cells follow, okay? So so what you see here are data for surface area and volume of E. coli cells, okay? There's actually 5,000 data points here. And these are data taken across different nutrient conditions, under different types of knockdowns, right? And under different antibiotic treatments. And we found that the best fit line is this, surface area is equal to 2 pi times volume to the power 2/3. This 2 pi was not coincident. We found that the best fit is like 6.28. So we put a number 2 pi, right? We still don't know what it means, like, well, why 2 pi? But but the exponent is easy to understand, right? And so if the surface area scales with 2/3, volume to the power 2/3, what it means is that a cylindrical cell, if it has such a scaling law, it means that they're conserving aspect ratio, okay? And they're conserving aspect ratio over 2 orders of magnitude, okay? Which is great, which is amazing. And I want to say that not all cells are purely cylindrical. They can be a little bit curved. Under antibiotic stresses, cells take a wide different variety of shapes. But it was quite interesting that all of them collapsed to this single law, right? So what this means is that if surface area scales as 2/3 of volume, then aspect ratio is conserved. If surface area scales with volume, which means that the width of the cell is conserved. If surface scales as volume to the power half, that means the length is conserved. Right? So you can come up with these different scaling laws. This is for E. coli. So we wanted to look at other organisms. Okay, so we looked at Bacillus subtilis, which is another very well-studied bacterium. It has a different scaling law. Okay, which is not 2/3, but it's 0.85, which means that it conserves aspect ratio like approximately. Okay. Yeah. So we it turns out that all different types of bacteria and even archaea follow this type of power law relationship between surface and volume. Okay. So we looked at 50 other organisms. Okay. And yeah. Okay. So what does this mean in terms of fitness? And that's something I wanted to get into, right? And so if we have such a law, what does it mean for the bacterial size and fitness, etc., right? So let's recall our favorite nutrient growth law. And here I'm just fitting it by an exponential. So we know that cell size increases with growth rate, and surface is 2 pi volume to the power 2/3. What that implies is the surface to volume ratio decreases with growth rate. Okay. So what this means is that if not a lot of nutrients are available, cells will try to increase their surface to volume ratio, right? And what that does is that it increases nutrient influx to the cell's surface, right? So there is an adaptive advantage of increasing your surface to volume ratio under low nutrient conditions, right? If you have if you're exposed to high nutrient conditions, you don't need too much food, right? So you can probably reduce your surface to volume ratio, conserve your energy, and stay fit. Okay. So right. So depending on the surface to volume scaling exponent, you can classify bacterium that are more adaptive versus less adaptive, right? And surface that scales with volume are not adaptive to changes in growth rate. But something like E. coli is highly adaptive, right? So they can transform their morphology to adapt to different media. Okay. So yeah, so this can be summarized using this figure here. So if you take a bacterium and expose it to maybe nutrient upshift or downshift, like more food or less food, right? On the nutrient downshift, surface to volume will increase, and on the nutrient downshift, surface to volume will decrease, right? Okay, so the sort of a hypothesis that we came up with at this stage is that if you inhibit growth of bacteria, they would want to reduce their surface to volume ratio. Okay, and that's advantageous because that can increase sort of the that can lead to faster growth, okay, because of this relationship. So sort of the hypothesis that we came up with. So we all know that antibiotics inhibit bacterial growth, right? So the hypothesis at this stage was cells can adapt to antibiotics by reducing surface to volume ratio, okay? And why would cells want to do that? Because if you reduce surface to volume ratio, you're actually importing less antibiotics. Okay, right. So that's a sort of a fitness advantage you get by doing so. Okay, so by just changing a morphology, you can be more adaptive to this type of drugs. Okay, so we wanted to see actually if that's the case. Okay, and are there any questions at this point? Sorry. Okay. Yeah. So
Sheila, can I ask a question? I'm going to be an avatar of some standard biologist that would say, the adaptive response in response to changes in nutrients or changes in antibiotic environmental stresses or whatever is gene expression. You reduce some pumps and this and that. So and there's some evidence that what they say is not completely wrong. So I think is it the case that what you're saying is that on the sort of shorter time scale, the adaptation could indeed be gene expression, etc. But longer term adaptation to these stressful conditions might be in these much more geometric morphological properties?
So that's a great question. So I will come to the timescale part in a moment. So indeed, these are long timescale changes, but
long timescale, Sheila, meaning multi-generational?
Multi-generational, yes.
Multi-generational, but still not evolution.
No, it's still not evolution.
Right,
but,
right.
I'll show the data in a moment. But what I wanted to say is that, yeah, so gene expression, yes. So gene expression would regulate protein synthesis, and that can affect shape. So we don't know if these morphologies are sort of active shape changes, or these are sort of results, right? Are these byproducts, or are these active shape changes? We don't know. We have to do careful experiments. But the hypothesis I'm trying to put forward is that morphological changes can lead to benefits. So at this stage, I don't know, but let me come to that in a bit. So,
sorry, just to make you repeat, why is that?
Why is what?
Yeah, so why does making, let's say, increasing your volume make you, and keeping your surface area constant, like why does that make it better adaptation?
Yeah, so let's take the case of an antibiotic, right? So antibiotics enter the cell through the surface, right?
Okay.
So if you reduce surface compared to volume, you reduce the effective influx rate of the antibiotics.
Right, okay, it's diluted more.
So you will dilute the antibiotic,
yeah,
right? By doing just morphological transformation, right? And so that's the idea, right? And this could be, yeah, purely phenotypic changes, right? Without talking about mutations, etc. And I mean, by manipulating, the cytoskeleton or whatever, you can sort of do this. So we wanted to look at data at this point. So what's evidence is out there? So we looked at lots of data, and what we found is that robustly, most cells that we have seen on the different types targets of antibiotics, so whether antibiotics target DNA, RNA, cell wall, ribosomes, they all reduce their surface to volume ratio. Okay, so by blue, you see cells that reduce surface to volume ratio, and the red ones are those increase surface to volume ratio. And yeah, so we found that by looking at the data, what happened? I think I lost my keynote.
Did we? No, Karna's still here. We're not seeing your screen share anymore.
I know. It's just I don't know what happened. So I'll start resharing it. Thanks. Oh, I need to go to. Okay, cool. So yeah, so so we did a little modeling of this by looking at how antibiotics come into the cell, how they bind to different targets, and we found that yes, indeed, that what's happening here? I think there may be some problem with that slide. Anyways, so we saw evidence of the fact that you have these shape changes, okay, in response to antibiotics, and mostly cells to reduce the surface to volume ratio. So we wanted to know, how does this happen dynamically, right? So this project actually started while I was still in Chicago, but I was about to leave, and I was working with Aaron Dinner at that time, and there was an undergraduate, Aaron, who was doing some experiments in antibiotics, okay? And the experiments were like this. So we were subjecting Caulobacter cells to antibiotics that target ribosomes, right? So these are ribosome inhibitory antibiotics, okay? So that means there's a global shutdown of protein synthesis, so cells grow much slower, okay? And so this is chloramphenicol, which is the antibiotic. And what we find is that the growth rate of the cell, okay, initially there is a very sharp decrease, right? But so in response to the antibiotics. So this is a step pulse. And then over time, over tens of generations, these cells adapt to that antibiotic, okay? The growth rate sort of comes back to its pre-stimulus level, okay? So this is a classic sort of classic example of adaptation, right? So there is some inhibitory stress and you overcome that stress, okay, over time, right? And okay, and what happens is that through this change, cells undergo drastic shape changes, okay? Cells not only increase their diameter of the width, which means that they reduce their surface to volume ratio, which is consistent with the hypothesis that I just talked about, but they also increase their curvature. So they become more and more curved, okay? Yeah, so irrespective of the concentration of the antibiotic, cells become more and more curved, okay? And yeah, in fact, if you can see at a much higher dose of the antibiotic, cells become almost C-shaped, right? So we thought there's a connection between shape change and adaptation, right? So in fact, Julie Theriault and Lee Harris did similar experiments, but not so much. But they had few data, and they also found that surface to volume undergoes reduction under addition of chloramphenicol, which is the drug that inhibits protein synthesis, right? So the model that we proposed was that this happens due to mechanical feedback, okay? And so the surface to volume reduction could be explained by advantage for reducing the influx, but to explain why cell curvature would change, that's not so simple, right? And to do that, we had to invoke mechanics, right? Which was missing from this whole picture. So let me just go through some simple explanation of what's going on, some mechanics, and then using that, hopefully I can explain why we see what we see, right? So here is a very simple explanation. So here's the underlying physics of cell shape and growth control explained using simple mechanics. So to grow a cell, you need an effective growth pressure, right? So this is not purely mechanical pressure, but you can think about an effective pressure due to growth, okay? So in fact, within a bacterial cell, you have osmotic pressure, etc. But I'm thinking about a more general effective growth pressure, which can come from the addition of new material. So pressure is any type of force that sort of grows the size of the cell, right? So there is an effective growth pressure that leads to expansion of the cell, and that is countered by tension in the cell wall, right? And so the bacterium maintain an anisotropic shape because it has a higher tension across than along the longitude, okay? So they have an anisotropic tension on the cell wall, which enables them to maintain an isotropic cell shape, right? So the rate of growth of a cell depends on the difference of these two forces, right? So how fast you grow depends on how much pressure you have and how the pressure is acting against tension, right? So the pressure and the tension balance vertically, so so cells don't actually grow along the short axis in a cell cycle, but they grow along the long axis because there is an imbalance of force, right? Yeah, so this leads to expansion of the cell horizontally. So what the antibiotic does, it reduces the effective growth pressure, right? because it reduces growth rate. And it does so by reducing protein synthesis. So the protein synthesis will mess up with chemical potential of inserting new material on the cell wall, so that there's an overall reduction in the pressure, right? And so if you reduce the pressure, what happens is that now there is an imbalance of forces on the along the short axis, okay? And that will lead to bending, okay? And the bending would relieve some of the stresses and can bring back the growth rate to its pre-stimulus level, okay? And this is explained using this sort of cartoon here. So the rate of growth of a cell depends on the difference between sort of this growth pressure and the mechanical stress on the cell wall. Now the antibiotic reduces the pressure, so there is a fast reduction in growth rate. But over time, what happens is that the shape relaxes so that the stress reduces by the same amount as the reduction in pressure. So overall, the growth rate becomes similar, right? To test this, so the idea that this is coming from mechanical feedback, you have to show that indeed change in curvature has correlation with change in growth rate. So that's what we found, that delta K is the rate of change in growth rate. And we found that it positively correlates with the curvature of a cell. So that as the cell is increasing its curvature, the cell is growing faster and faster, right? So by this mechanism, we found another mechanism by which cell can actually change the morphology to adapt to growth, okay? So what I told you so far was a qualitative model. We actually built sort of a dynamic model of sort of the morphodynamics of a cell, how it depends on the growth rate. Do we have sufficient time? Maybe I can skip it. Yeah, what do you suggest?
We have time. I mean, how long are you thinking, Sheila?
A few slides, but
yeah,
maybe can, okay.
No, that's the whole point. The whole point is, if people are busy, they can leave. But for those of us that are interested, please continue.
Right. So this was sort of the basic physics underlying the growth control. Now we wanted to put this in mathematical language. So we developed a whole scheme of describing non-equilibrium shape evolution of cells, and this is based on sort of writing down dynamic equation of motion for the degrees of freedom of the shape, right? So for example, for the shape that we're interested in, which is Colobacter crescentus, the degrees of freedom are, you can think about the degrees of freedom as the radius of curvature, the width of a cell, which is variable across the long axis, and also the opening angle of the cell theta, right? And then you can formulate an equation of motion, which I'll not get into the details of how we got there, but the idea is that the rate of growth depends on the driving force, okay, which is given by the gradient of energy, right? And so we have to construct some energy functional here, and the energy functional depends on a few things. So we are talking about the growing system. So we have to account for growth, okay, which is taken into account by this term, where epsilon is a chemical potential and A is the surface area. So this term describes growth of the system. And this growth competes with strain energy, which is responsible for maintaining the shape. There's a bending energy for maintaining width, and there's a bending energy for maintaining cell curvature, right? So you can put this in a model and evolve the model. And the model actually does, and this is the scheme of the model. So it's a mechanochemical model of growth. So first of all, the mechanics determine shape. The chemistry determines how you add material on the cell surface. And then based on conditions such as adder principle, you make decisions, right? So cells make probabilistic decisions whether to divide, right? Whether to progress the cell cycle, or whether to alter their mechanics depending on stresses, right? So if you have antibiotics, so it can change its mechanics because it changes the effective growth pressure of the cell, right? So And this is the scheme, and we started working this at UCL. So Roshan was my PhD, an undergrad who immensely helped with this simulations. So the model turned out to be very successful in the sort of describing the dynamics of the cell. So using this, we could capture not only the adaptive behavior of the growth rate and division time, cell cycle, but also cell shape, right? Okay. And then not only to sort of steady changes, but also cellular adaptation to fluctuating concentration of antibiotics, right? And that's another experiment that was done. So the model predicted that cells will adapt to fluctuating concentration, and as a result, what they will do is they will do reversible shape changes, right? Because this is a mechanical feedback, right? So the mechanics could be reversible, right? And so that's what we saw, that as the cells were exposed to stress, the curvature and the width would increase, right? And as you remove the stress, the curvature and width will go down, and it was reversible. And this and experimentally, we also saw this sort of shape oscillations that accompanied this adaptive response, right? One of the interesting things that you see in experiments, which is not fully captured by the theory, is that there is some sort of learning, right? And we see that upon repeated exposure of stress, cells actually inhibit growth by lower and lower amount, right? So there's actually some form of dynamic learning, which is not captured by this model fully. And that's something I think leaves room for future work, right? So how do cells actually recognize fluctuations, like fluctuating environments, and sort of learn from it, okay? So I think that's an exciting area of sort of open work. Okay? So overall, I wanted to summarize this whole section by this sort of figure. So to have adaptation, you need some underlying feedback control, right? In systems biology, we have a lot of biochemical feedback control. Here, I'm talking about a mechanochemical feedback control, which works like this. So there is an input stimulus, and there's an output activity, which is the growth rate, right? So the output activity initially goes down, right? There's a rapid reduction, but over time, it sort of comes back to its pre-stimulus level, which means there is some sort of a memory variable in the cell that is giving some negative feedback control, right? Which enables this to come back. So in this case, that control element is cell shape, right? And cells can change their radius of curvature and surface to volume ratio to sort of give that negative feedback control. But there could be other variables, right? There could be, so for example, under stress, like cells can produce more ribosomes. So if you inhibit ribosomes, cells can actually make more of them. And you can get a similar response, right? So, so yeah, changes in gene expression could also lead to similar things. So what we need to do is a careful study of different types of models and sort of rule out one versus the other, doing careful experiments, right? Yeah, so I think these are not done, but I think this leaves a lot of room for future work. And I just wanted to leave with a few open questions. So first of all, yeah, so I think the whole question about how morphology regulates adaptations to stress is an open question. We have some theory and some experimental evidence, but I wouldn't say that we have proved one way or the other, right? So this is an interesting idea that morphological changes can give you some benefit, adaptive benefit, but it could as well be a byproduct of some underlying biochemical change, right? Rather than being an active change, right? So that's something we need to disentangle by doing good experiments, right? So for example, I mean, if you engineer bacterial strains with more curved morphologies, are they more resistant to drugs, right? So something like that, right? So if you actively change shapes do you get more things out of it. And the other question I wanted to get at, yeah, Felix, I'll come to you in just a second. Let me finish like 2 sentences, then I'm done. So I started with a proteome allocation model of cell size and growth control, how cells allocating different resources. And in the end, I was talking about a different sort of energy allocation model, like how cells are allocating their energies for growth, shape maintenance, metabolism. These 2 pictures have to be connected somehow, right? Now, I think we don't know. So some work needs to be done there, like how to connect to the mechanics part of the cell with the underlying proteome, okay? And then I guess like understanding physiology in fluctuating environments is an open area, right? Where a lot of work needs to be done, okay? Yeah, thank you so much. And I would like to end with this slide, especially thanking Diana, Nicola, and Roshan, who did a lot of work. Clevin Lowe did these experiments on antibiotics. Thank you.
Thank you so much, Sheila. And yeah, we can go ahead with Felix's question, I guess. If you want to unmute yourself.
Hi, thanks for the talk. I really enjoyed that. So I had 2 questions actually. So first off, I was interested in the hypothesis that the rounding out during chloramphenicol treatment could be a adaptive response to reduce the internal concentration of antibiotic. And I guess I was wondering if you've done any back of the envelope calculation to estimate how much of a change you would expect to see in the internal antibiotic concentration as a result.
Yeah, we have this bioarchive paper where we estimate the antibiotic dilution, and you can go up to 50% dilution, depending on, so it depends on several things like antibiotic biochemistry, what is the rate of import of the antibiotics, right? So it's not that simple, but we computed a number, and that paper is under review right now. And so, yeah, we find a range from around 20% to 50%.
Oh, that's actually quite large. That's more than I would have necessarily thought. So that's interesting.
Yeah.
My second question was in the latter case where you were thinking about the adaptive shape response to antibiotic treatment, you said that it was started by the internal turgor pressure reducing. But I guess I was curious about how the antibiotic would be doing that, given that much of the turgor pressure in a cell is generated by small ions.
Right. So I think I was very careful to say that this is an effective pressure, not only the turgor pressure, right? I don't think the chloramphenicol is affecting turgor pressure directly, although we have evidence, like there are some evidences of chloramphenicol affecting turgor pressure. But here I was just giving a sort of a mathematical argument where, the pressure could come from 2 sources, a mechanical pressure, which is the turgor pressure, and you can think about a chemical pressure, which is a sort of force that grows the cell, right? And that chemical pressure comes from addition of new material to the cell wall, right? Which is synthesized from within the cell. That is what is reduced by chloramphenicol, not the mechanical part, okay? To put it more rigorously, what chloramphenicol does, it changes this parameter epsilon, okay, which is the chemical potential for adding surface area material, right? The cartoon that I had earlier was more like a sort of a, toy model explanation for what's happening. Right.
Do you have an idea of how the change in like that epsilon constant would be, initiated by chloramphenicol? It's kind of interesting.
Yeah, so I imagine that, so epsilon regulates the rate of addition of cell material, so cell surface material, and that is controlled by different types of proteins, right? One of them is MREP, right, which directs cell growth. So what chloramphenicol does is reduces protein synthesis globally. So this process will slow down. So epsilon is an effective parameter here, which regulates synthesis of cell wall, which is regulated by lots of other proteins. So yeah, so by reducing protein synthesis, plant funicular effectively reduced epsilon. And we have computed how epsilon depends on plant funicle concentration in the paper. But the sort of detailed biochemical pathway, I wouldn't be able to tell you. Okay.
Cool. Well, thank you very much. That was really interesting.
Okay, thank you.
Can I ask another question about the epsilon, just so the growth pressure?
Yes.
So would you get anything different if you had like a different pressure, or that you had like growth that was different along the different curvatures? Sort of like.
Yeah. That sounds complex. Yeah. You're talking about anisotropic growth?
Yeah. I guess if it's like sort of other than isotropic.
Yeah, I guess so. You can get different types of morphologies, surely, using that. That's a more complex model, and I think there are some evidence for anisotropic growth. So yeah, we are not currently working on that, but that sounds interesting. Yeah.
Thank you.
Thank you.
Sheila, a silly question surely, but I'll ask it. If one looks at sort of diversity of cell morphologies in some sort of section of the tree of life, so nearby species, nearby bacterial species, and I know there is some variation in morphology.
Yeah.
Are you at all inclined to suggest that?
This is the slide. Sorry, I just wanted to show you the slide. I have a slide for morphology.
Okay, well, are you at all inclined to suggest, am I right that the wild conjecture based on your hypotheses, which are very appealing, is that the actual stressor conditions that these cells have adapted to, they're sort of deterministically different stressor conditions, not just fluctuating environments, but they're really in different stress conditions, and their morphology has therefore adapted over much longer timescales to confer that ability to respond. Is that the hypothesis?
Yeah. I mean, I would say so, because at the molecular level, some of these are not very different.
Right.
So E. coli and Colobacter, so which is like, let me give a simple example. Colobacter is curved, E. coli is rod-like straight. They have very similar cytoskeleton structure, etc.
Right.
Everything. But I think so. This is a result of adaptation to environment, what environment they have been living in, and I think that's true for all forms of life. So, and yeah, I would agree with that. I don't know how to prove it, but
I mean, you're circling around something that Karna and I have been, and various of us have been talking about, which is this idea that the kind of physiological adaptation of a species says something about the possible evolutionary adaptive paths that it has access to.
Yeah.
Those two sort of statistical ensembles might have something to do with each other.
That's right.
And that's I've tried to sort of assess that in very different systems, different scales, but yeah, it's a very appealing hypothesis that adaptation at short timescales and long timescales have something to do with each other.
Yeah.
Beautiful.
I agree that.
Beautiful.
Yeah, that was really cool.